structure. So here, for example, two ring-like structure is present. So that means one structure is like that. That means 13 protofilaments are present. And to that again one, for example, this is the one tube, and to this another tube are joined. Same like structure. So that means if we take the cross section, it looks like this. That means one, another structure. So this is the main structure of like this. So this is called the peripheral ring. So in the exonym, the outside ring are called the peripheral ring, and they have 9 peripheral rings. This is the first, this is the second, this is the third, fourth, fifth, sixth, seventh, eighth, and the ninth. So that means 9 protofilament-like structure, the 9 microtubule structure is present in the peripheral, and in the central region, this is the central region, this is the central region, or it is called as the central ring. And in the central region, 2 structures are present. One, another is a 2. But here they are present like in the diploid structure or the doublet structure. This is the doublet structure. So the exonym structure, as we, or if we look the structure, the exonym structure, it is designated as 9 plus 2 arrangement. What is the 9 plus 2 arrangement? So 9 means in the peripheral structure, 9 structure or the 9 microfilament structure is present, and which are the doublet. But here the 2, that means the single structure, and that is present in the twice. This is the one single structure, this is the another single structure. That means 2 single, that means it is the 2 singlet. So that means 9 doublet and 2 singlet, and the 9 doublet are present in the periphery region, and the 2 singlet structure are present in the central region, and the all overly it is called as the 9 plus 2 arrangement. It looks like this. So now this is the central region, and in the central region the central link is joined to the peripheral link by this structure, and this structure is called as the radial spoke. So that means the radial spoke, it joins to the peripheral link to the central link, while the peripheral links, they are again joined by the connectors. These are by connectors. So these connectors are called as the nexin. So these are the connectors, or they connect the peripheral link structure. So now to this peripheral link structure, a structure like this structure is present. Here the 3 dot, if we series, this 3 dot structure, this is main important structure, that is called as the dynein arm. So this dynein arm, it helps in the, or it releases the energy for the movement of the, for the movement of the organism. So that means for movement, some energy is required, and this energy is released by this dynein structure. Structure. So this is the all about the exonym structure. So now come to another structure, that is called as the basal body. So that means, for example, this is the paramecium, this is the cilia structure, and this inside the body or in the cytoplasm structure, the centrioles are present. They are called as the centrioles. So now in the basal body, the centrioles forms the cilia and the flagella, and the process is called as the ciliogenesis. So what is the ciliogenesis? So that means inside of the body, or the inside, not our body, that means inside the, for example, the paramecium or the euglena. So this structure, so here the centrioles forms the cilia and the flagella, and the process is called as the ciliogenesis. So during the ciliogenesis, the centrioles are called as the basal body. So this is the again another structure of the cilia and the flagella which is present inside the body or inside the cytoplasm. So now the structure of the basal body is different. Look at the structure, and this is same as present in the centrioles. So first, before going to the basal body structure, first we should know something about the centrioles. So what is the centriole? So for example, in the cell or during the cell division, for example in the metaphase stage, they are the chromosome which is present in the metaphasic plate. So now the spindle fibers are formed here. So this structure is called as the centrosome. So now what is the centriole, what is the centrosome? So now if we see the structure, the centrosome, here it looks like this. So this is the centrosome. So in the centrosome, two centrioles are present, and they are present perpendicularly to each other. So they are called as the centriole. So in one centrosome, two centrioles are present, and they are present perpendicularly to each other here. And besides this centriole, some amorphous structure is present, and they are called as the PCM. PCM means pericentriolar matrix. Just imagine, just like the cytoplasm. So that means inside the centrosome, two centrioles are present, and they are present perpendicularly to each other, and now the PCM or the pericentriolar matrix are present. So now if we look at the centriole structure, let's suppose this is one centriole. So in the centriole, the structure looks like this. Just imagine. So that means in the axoneme, two structures are present like this here. So that means, for example, this structure, that means one tube and it is joined to the another tube. If we take cross section, it looks like this. So here, three tubes are joined to each other. 1, 2, 3. So just like that, 9 peripheral structure, 9 peripheral structures are present. So in the axoneme, this is the axoneme structure. So now in the basal body, it looks like this. That means this is the axoneme structure, or this is the basal body, which is similar to the centriole. Okay. So in the basal body of the centriole, the same structure like present, but what is the difference in the axoneme? So the peripheral link are called doublet. So they are the doublet. That means they are present two ring structure, but they are the triplet. So that means they are present on the peripheral ring, same, it is called as the peripheral ring structure, and this is called as the middle part, they are called as the central ring structure, or central ring or central structure, it is also called as the hub. So in the peripheral ring structure, the triplet microtubules are formed, while in the axoneme structure, the doublet microtubule structure are found. And another difference is that inside the central region, there is no microtubule structure. But in the axoneme structure, they have two singlet structures are present. So that's why the axoneme structure is called as the 9+2 arrangement, and the basal body or the centriole is called as the 9+0 arrangement. And here the difference is the 9 is the 9 are the doublet, and here the 9 are the triplet. This is the main difference between the axoneme structure and the basal body structure. So now look at the structure here. So it looks like the bullock cart wheel structure. That means in the, let's suppose this is the bullock cart, this is the one wheel, and inside it another wheel-like structure. So here the centriole is also looks like same as bullock cart wheel structure. So that's why the centriole is also called as the bullock cart wheel. This is the very, very important, and in the previous exam this question was arised that which structure is also called as the bullock cart wheel structure, and the answer will be the centriole. So this is the all about the structure or the difference between the axoneme and the basal body. So the basal body, it is same as the centriole as in the basal body structure. Here the centriole forms the cilia and the flagella, and the process is called as the ciliogenesis. So now this is the centrosome, this is the another centrosome. Here in the centrosome, two centrioles are present perpendicularly. Here also two centrioles are present perpendicularly. So now actually first in the cell one centrosome are present, but for example in the cell cycle, this is the M phase, this is the G1 phase, this is the S phase, this is the G2 phase. M phase means division phase, that means mitosis or meiosis. G1 means this is the gap 1. This is the S phase, that means here the DNA is synthesized, or the DNA becomes double. So now this is the G2, that means gap 2, that means the gap between mitosis or the meiosis and to the synthesis phase. So during the gap 2 phase, so now the centrosome becomes the double. So the centrosome, actually in the cell, the centrosome are present 1. The number of the centrosome are 1. But during the gap 2 phase, the centrosome becomes double, just like the binary fission. So in the gap 2 phase, it becomes the double, and now it comes to one end and another is come to another end, and now the mitosis and the meiosis occurs. So now the spindle fibers are formed. So this is the all about the centrioles or the centrosomes. So now come to what is the function of the cytoskeleton. or what is the function of the microtubule? As you have already discussed, but just as a summary I am telling you that one is the spindle fiber formation. The second one is the formation of axoneme and basal body of cilia and flagella. Third one is the it also helps in the cell differentiation. So that means which cell should convert to which cell, and it is determined by the microtubule. Another is the it also distributes to the cell pigment. That means, for example, the chloroplast or the melanocyte. And another is that it also helps in the cell movement. That means during the gastrulation, one cell goes to another, or one cell again come to here. So this is the cell movement, and this cell movement is occurred by the microtubule, and most importantly, it provides the support to the cell or to the organisms. So this is all about the structure of the microtubule. So if we like the video, then please like and subscribe my video. Thank you. The second cytoskeleton is microfilament. So microfilament. So now the microfilament, here the component of the microfilament is actin plus ATP. Actin plus ATP. But in the microtubule, the main component was tubulin plus GTP plus magnesium. But here importantly GTP is present, and here ATP is present, and here tubulin is present, and here actin is present. So this is the main difference between the structure of the microtubule and the microfilament. And you can memorize like here A for actin, A for ATP. If you confuse between the ATP and the GTP. But importantly, ATP is present in the microfilament and the GTP is present in the microtubule. So now, where the microfilaments are found? So firstly, it found in the muscle cell. In the muscle cell, that here, besides the actin, another myosin is present. So myosin along with actin, it forms the myofilament, and this myofilament helps in the muscle contraction or muscle movement. So the muscle contraction is provided by the microfilament, and along with that it also helps in the, or it is present in the microvilli. The microvilli, that means in the small intestine, a folded part is present, and that is called the microvilli, and it helps in the active absorption of the food in the small intestine. So it also be provided by the microfilament. So now main functions of the microfilament are, one is it helps in the locomotion. So locomotion means it is present in the pseudopodia. That means in the amoeba case. So here one pseudopodium. Pseudopodia is the plural. The singular is the pseudopodium. So here the pseudopodia, so the microfilament is present and it helps in the locomotion. The secondly, it also helps in the muscle contraction. Muscle contraction. So now third one is it helps in the support to cell. So remember please, all the cytoskeleton, that means microtubule, microfilament, intermediate filament, they have the main function is the support to the cell. That's why the name is the cytoskeleton. The skeleton is for providing the support to the cell. So now it also allows the intracellular movement. Intracellular movement. Intracellular movement means, suppose inside a cell, suppose this protein and it wants to go here. So it can be occurred only by the microfilament. That means microfilament, it helps in the movement of the anything inside the cell. That means it can move the protein, it can move the carbohydrate, it can to move the lipid. So it helps in the allow the intracellular movement. Along with that, it also helps in the streaming movement. Streaming movement. The streaming movement, it is also called as the cyclosis. So what is the cyclosis and the streaming movement? It is simply the movement of cytoplasm. Okay, so the movement of the cytoplasm or the cyclosis or the streaming movement is occurred by the microfilament. And another is that it also helps in the cleavage during cell division in animal cell. So that means during the cell division, first the karyokinesis occur, karyokinesis, and another is the cytokinesis. Karyokinesis is the division of the chromosome. It occurs first. Then the division of the cytoplasm, or called as the cytokinesis occurs. That means at the middle, the cleavage occurs, and this cleavage is due to the microfilament. So these are the main function of the microfilament. So now come to the intermediate filament. Intermediate filament. So unlike the microtubule and the microfilament, intermediate filament, they don't have many functions. They have only one function, that is providing mechanical support to the cell. Or cell support, it is the main function of the intermediate filament. For example, the most example of the intermediate filament is the keratin. Keratin, it provides the support. Again, the laminin, it also helps in the support to the cell. So these are the main function of the intermediate filament. That means it only provides the mechanical support to the cell. So these are all about the cytoskeleton, that means microtubule, microfilament and the intermediate filament. If you like the video, please like and subscribe, and tell others to subscribe also. Thank you.